This is Dr. Chithrani from University of Victoria. We use gold nanoparticles as a radiation dose enhancers in cancer radiotherapy. We use confocal imaging and TM imaging to probe gold nanoparticles in cancer cells. However, we find that this CytoViva hyperspectral imaging technology is very user-friendly and also less time-consuming if we were to look at gold nanoparticle within cancer cells. For example, this image shows a cell where nanoparticles are not targeted into the nucleus. So you can see there is no signal coming from the nucleus, but in this case, the nanoparticles are targeted into the nucleus. So all these bright spots are basically nanoparticle in the nucleus. We can also verify whether these bright spots are gold by taking a spectrum in that spot. So we know for sure these are gold nanoparticles. We also look at not only at a monolayer level, like two-dimensional, we were able to look at the nanoparticle in tissue structures as well. All these yellow structures are gold nanoparticle within tissues. We move forward into animal studies where we look at animal tissue sections to identify gold. For example, this is actually a blood vessel where you really can see gold nanoparticles in the blood vessels, and also within the tissue also you can see gold nanoparticles. So this is a very versatile technique to probe gold nanoparticles in cancer cells. Thank you.